the effect of UVC radiation on the germination rate, time till germination, and growth rate of radishes, Raphanus sativus, by George James Montague. Introduction. There are three different bands of UV radiation, A, B, and C, which are divided by wavelength. UVA has the longest wavelength from 400 to 315 nanometers. UVB is in the middle, with 315 to 280 nanometers. UVC is the shortest, from 280 to 100 nanometers. In addition, shorter wavelengths carry higher amounts of energy, but aren't able to penetrate through solid matter as efficiently or effectively. And this is seen in Figure 1 with the three bands of UV radiation, UVA, UVB, and UVC. And on the x-axis is a photon, or the wavelength, and on the y-axis is the amount of energy it carries. You can see that UVC carries substantially more energy than UVB and UVA. Peak absorbance is a wavelength at which a molecule absorbs energy of photons most efficiently. DNA has a peak absorbance at around 260 nanometers, and protein and lipids are at around 280 nanometers. However, this varies from the exact protein and lipids as their structure, and therefore peak absorbance does vary a bit. Cellular death and dysfunction, as well as genetic damage, are all common results of UV exposure of both A, B, and C. My research question was, how does exposure to UV radiation and UVC radiation in specific affect the growth rate, germination rate, and time till germination in radish seeds, or sativus? My hypothesis was, if there's an increase in levels of UVC radiation exposure, then there'll be a decrease in their germination and growth rates and an increase in the average days until germination. Methods. The research was conducted at my school, St. Paul Academy and Summit School in St. Paul, Minnesota. And there are two groups of radishes, or sativus seeds, which received exposure from a 25-watt, 254 UVC bulb. And the 254 nanometers emitted from the bulb is very close to the 260 nanometers of the peak absorbance of DNA. One of the groups received 5 minutes of radiation, which resulted in 7.5 kilojoules per meter squared, and the other was for 10 minutes, which resulted in 15 kilojoules per meter squared. There was a total of 100 seeds planted, 50 for each group, 50 for high radiation, 50 for low radiation, as well as 50 seeds as control, which received no radiation. Seeds were grown in germination trays with cotton strings acting as an irrigation system. This is seen in Figure 2, which shows the irrigation system of the cotton strings, with the cotton strings coming from the bottom of the container where the water reservoir is. So each plant gets the exact same amount of water, and there is no water transfer between different containers of plants. Figure 3 shows the completed lab setup, which all the plants planted in their individual trays and ready and watered. Plant height plus or minus 0.5 millimeters germination data was collected every 3 days over a 35-day period from November to December of 2020. Air temperature, watering, and light were kept constant at the study, and when we ANOVA, as well as 2-tailed t-tests for use as statistical analysis for the data to find significance where P was less than 0.05. Figure 4 shows the radishes, in specific, the high radiation radishes, after 35 days of growth in the included growth period. Results. A positive relationship was found between high levels of UVC radiation and the germination rate. Germination rate. The 10-minute group had significantly higher germination rate than both the 5-minute and the 0-minute groups, 2-tailed t-tests where P was less than 0.05. There was no significant change between the 5-minute and the 0-minute groups, 2-tailed t-test where P was greater than 0.05. Mean time till germination. There was no significant difference found between the 10-minute, 5-minute, and 0-minute groups. ANOVA or P was greater than 0.05. And as seen in Figure 6, the 10-minute group had a more normal distribution that's seen in the blue, whereas the other groups had a higher percentage germinate on one side, specifically on the left side in the first 3 days. This is seen with the yellow or orange and the red, where the blue has a more normal distribution. The 10-minute group and the 5 and 0-minute groups had a more of a lopsided germination distribution. Growth rate. There was no significant difference found between the mean growth rates of the different variable groups. ANOVA, where P was greater than 0.05. In addition, for the growth rate, growths were taken after germination so that they were standardized and that there was not a difference in growth rates for how long it took the seed until it germinated. Discussion of future studies. The hypothesis of the study was not supported. High levels of radiation have a positive impact on the germination rate, not a negative one. High energy UVC rays may have caused damage to the seed coating, lipids, and proteins, or cause genetic damage. A weakened seed coating will allow for water to fill the seed faster and for the embryo to break the seed coating easier, which would have allowed for higher germination rate as more seed coatings would have broken. But the seed coating would have blocked the rays from penetrating further into the seed and doing actual genetic damage, which would have changed the growth rate, which was not observed. Fewer seeds were needed as a result from the higher germination rates, which is more cost-effective planting for farmers as they'll need to plant fewer seeds and buy fewer seeds, as well as less organic waste and better water conservation as water and fertilizer would not be used on seeds that would never germinate, which again, not only is better for the environment, as runoff is a big problem on farms and goes into the ocean called algae problems, but also it helps farmers as they have to buy and pay for less water and fertilizer. It also allows for easier transportation of seeds, both on the terrestrial scale, so for transporting seeds to different farms in large batches, and also on the interplanetary scale for long-term terraforming of their planet and agriculture. Seeds need to be transported, and it's not sustainable to keep transporting seeds back and forth. And so by allowing some radiation and fewer seeds, it'll be easier but to transport them as they would be cheaper as you don't have to have shielding to them, and also because only fewer seeds need to be brought as there is a higher germination rate. More research is needed on the adverse health effects of the higher radiation dosages, however, as it was inclusive in the study as genetic damage was not found. Additional research is needed on genetic damage and implications caused by the irradiation of seeds because although there was no significant difference found in the growth rate, this study did not assess the actual damage to genetics and DNA, and that was not looked at. So more studies need to be done on that. For acknowledgement, I'd like to give special thanks to Dr. Gary Garner of the University of Minnesota and Dr. Gioia Massa of NASA for the guidance for the development of this project. And this is my bibliography. Thank you.